can do this. If you have questions, methodological questions that we're not able to get to at the end of the talk, please feel free to note down my email address. You're welcome to shoot me an email. We can talk later on. Anybody that's potentially interested in doing graduate work here at BYU, also let me know. I'd be happy to talk. So let's jump in, and we're going to talk about your favorite group where you know there are taxonomic problems, right? Whichever group that is, take a minute and ask yourself, if you could choose one group with known taxonomic or evolutionary issues, which would you choose? And once you select that group, ask yourself, well, why are there taxonomic problems? Is it because that there's a wide range of variation, phenotypic variation in that? Is it because there are interesting biogeographic patterns in the distributions of this taxon? Maybe it's simply because genetic data have suggested that there are issues with those groups. But we know that taxonomically problematic species level or species level groups with taxonomic problems are rampant across the at least 20,000 or so lichen species that have currently been described. So knowing that you have a favorite group with taxonomic issues, what are some of the things that you would want to do to try to address those problems in your specific group? Okay, so hopefully you have something in mind. Again, what is your favorite taxonomic group with problems, and how could you try to address those problems? As we've thought about this, here we're focusing on Lecanora polytropa, but I think the issues that we deal with are probably much broader than just a specific taxon. So as I asked myself this with polytropa, the things that I came up with in terms of what we need are, we need appropriate specimen sampling, both across the distribution, the geographic distribution of this group, and also across the taxonomic variation. Within Lecanora polytropa, there are a number of described forms and subspecies. Can we effectively sample all of those? In addition to good specimen sampling, we also need to consider effective and appropriate morphological and anatomical sampling and characters. There's issues in these groups, despite the fact that we're using, phenotypic traits. So perhaps the traditional characters that we've used to diagnose diversity here may not be appropriate, and we may need to look beyond those traditional taxonomic features or characteristics that have usually been used to try to characterize this diversity, and look outside of those. I love using sequence data, so of course on my list I have, we need good genomic sampling. Can we just use the formal or the standard fungal barcode, the ITS? Would we need additional loci, maybe a multi-locus study, or can we use genome-scale data to help resolve some of these problems? And all of these questions are based on how we conceptualize what a species of lichen-forming fungi is. Are we okay with the general lineage concept that basically says, hey, any line of evidence that supports the evolutionary independence of this metapopulation lineage is sufficient to delimit that as a species? And are there coinciding empirical methods that allow us to delimit those boundaries? How do we integrate our genetic data or genomic data with that phenotypic data? Okay, so these are some of the issues that we're facing. And when we come to the Lecanora polytropa group, we can see, hey, it's found worldwide. We need to come up with effective specimen sampling strategies across its distribution. We know that there are a number of formally described closely related species. Can we sample those? We know there are a whole bunch of different varieties and subspecies. Should we sample those? And how do we start dealing with this for the Lecanora polytropa group? And this is a smattering of specimens that I've collected here in the Intermountain West of the United States. Lecanora polytropa, it was first described in 1791. Within this taxon, there are 23 formally described forms, 4 subspecies, and 24 varieties. Now, some of these forms and some species have been elevated to species rank or have moved out of the polytropa group with additional data. But this is an incredibly diverse group of fungi that has been lumped in together as Lecanora polytropa sensulato. There are a number of other formally described species. One of interest here on the right is Lecanora fuscobrunnea. This is an Antarctic endemic that's morphologically deviant from most Lecanora polytropa, but we know it's a part of this species group. And here in this list on the right, I have listed a number of other taxa that are also potentially a part of this species complex. Right, so looking at this, we know that there's a lot going on, and how could we deal with it? Well, let's start with specimen sampling. Like I mentioned before, our work has been focused largely on alpine and subalpine habitats in Western North America in a response to helping land managers understand how to characterize and deal with the diversity in these forests or these habitats. The site shown in a yellow triangle in the center on this map, this is again a map of Western North America with California here along the coast, the state of Utah shown here, and other states in the West. The yellow triangle represents an isolated sky island on the Colorado Plateau called the LaSalle Mountains. Most of our sampling occurred on that one isolated mountain range. We have a number of other samples from the Uinta Mountains shown with this red triangle, and then a few others from the Beartooth Plateau in southern Montana shown with the blue triangle, and then a smattering of other specimens we collected in high altitude forests and habitats throughout Western North America. So what we did is we went out and collected as many polytropa that we could, tried to identify the range of variation, and from that range of variation, we aimed to extract and amplify, extract DNA and amplify the ITS region, the fungal barcode, align those data, and come up with candidate species using the recently proposed ASAP species delimitation approach to come up with species partitions. And from those species partitions inferred from ITS sequence data, we wanted to do a validation or a concordance analysis by generating genome-scale data to see if the genome-scale data supported those same species partitions that were inferred from the standard fungal barcode. So we did some de novo shotgun sequencing, creating Illumina libraries and sequencing on a high-seq. From those short reads, we assembled draft assemblies using spades. And then from those draft assemblies, we pulled out regions of interest using genome skimming with the program Hive-Piper. Specifically, we pulled out known single copy fungal loci or protein coding markers. These are called the protein coding or the BUSCO markers. And from these, we generated an alignment of over 1.8 megabases. We also looked at the mitochondrial genome, and we were able to generate an alignment spanning the vast majority of the mitochondrial genome for Lecanora polytropa group with an alignment of over 65,000 base pairs. Right, so we came up with some cool data spanning from the fungal barcode, ITS alignments, to genome scale data for both the nuclear and mitochondrial regions. And from these, we implemented a number of species delimitation analyses. So let's dive right into the results, right? So with the ITS or the standard barcode, fungal barcoding marker, we generated sequences from over 128 specimens. 87 of those were newly generated here, and the ASAP species delimitation came inferred between 28 and 35 different species. Taking a look at this ITS phylogeny here, the branches shown in gray indicate the specimens from GenBank. The specimens shown in different colors represent the branches shown in different colors represent specimens collected from distinct mountain ranges in Western North America. right? So as you can see, looking at this tree, the branches in gray often do not include colored branches, indicating that the sequences generated from Western North America for this study are highly dissimilar or largely dissimilar from those sequences representing Lecanora polytropa that are currently available on GenBank, right? One interesting fact to look at is this group shown in green. These are specimens from Antarctica, the Lecanora fuscobrunnea group, and some of our specimens came out to be closely related to that. So I'm going to dig in a little bit deeper. Specifically, I mentioned the LaSalle Mountains in southern Utah. This range is isolated, and from this one single isolated mountain with limited alpine habitat, we inferred up to 14 distinct species partitions based on ITS sequence data alone. There's some other interesting features of those species partitions inferred from the ITS sequence data. Two of those partitions, distinct partitions, occurred in Western North America and also in the high Arctic. In contrast, one of those 35 species partitions occurred in Western North America and also in the dry valleys of Antarctica. And this was the species partition that included Lecanora fuscobrunnea. So we see these striking and fascinating distribution patterns where the 3 of the 35 cases where species partitions overlapped with our GenBank and our newly generated sequences, they have these really fascinating polar distribution patterns. So, all right, so there's up to 35 species in Lecanora polytropa. We have these wacky distributions with species partitions occurring in Western North America and the Arctic, or Western North America and Antarctica. Is this real? Can this even be real? So as we tried to complement the inference from the ITS sequence data, we turned to this genome skimming approach. And here we have our 2 phylogenomic trees. The one on the left is inferred from the Busco markers. This is a nuclear phylogeny inferred from 1.8 megabases from the nuclear genome. The tree on the right is from the mitochondrial genome, over 65,000 base pairs from that. And the tips, the boxes surrounding each tip, represent the ITS, the candidate species inferred from ITS sequence data. And we can see that those candidate species inferred from ITS sequence data were consistently recovered as monophyletic in both the nuclear phylogeny and the mitochondrial phylogeny, providing pretty compelling evidence that that inference of high levels of species diversity based on the ITS region are supported by genome scale data. That in fact, Lecanora polytropa likely has dozens of species, and we could only generate sequence data from those specimens that we collected from our own research. And when we use the species delimitation program SODA implemented in Astral, we see that in fact it supports at least as much diversity that the ITS sequence data suggests, and potentially even more. Our SODA analysis suggested up to 28 species simply on our specimen sampling in Western North America. So yes, in fact, it looks like there is tons or a ton of undescribed species diversity in Lecanora polytropa. So looking at it from a morphological and anatomical perspective, and we also looked at it using thin layer chromatography, it's looking like it's really, really difficult to come up with consistent diagnostic features that separate out these lineages. In the case of spore sizes, there can be some general trends, but they often overlap. There's no consistent diagnostic chemical features that distinguish or separate the species from Western North America. So we're left with this really messy perspective of, yes, we know there's tons of diversity in Lecanora polytropa. We know that there are fascinating biogeographic patterns, but how can we practically identify those without using sequence data? So as we kind of conclude this, we asked, what do we need, right? We need appropriate specimen sampling. For this study, we only looked at specimens from Western North America, coupled with sequence data, ITS sequence data from GenBank. We need to sample more broadly. We need to sample more broadly, both geographically and taxonomically. We know that, at least based on these data, there's limited number of specimens per candidate species. So to characterize those morphological and anatomical features, we need to increase our sample size. Genomic sampling, if we want to effectively infer relationships, we can easily do that from high throughput sequencing. But the fungal barcode is probably going to be a pretty accurate approach for characterizing candidate species level diversity. And then finally, integrative species delimitation. How do we integrate these phylogenomic results with what appear to be really messy phenotypic traits without any clear diagnostic features? And with that, I'd like to end and thank Jeffrey Clancy for all of his work. He's the co-author on this, but he's the brains and the brawn behind this work, the IAL committee for putting on such great meetings, Lecanomics project, and then some financial support. Great. Let me know if you have questions, and I look forward to talking to you.